بسم الله الرحمن الرحيم. أعزائي طلبة المرحلة الأولى، أبدأ معكم المحاضرة الرابعة من محاضرات الدهون. The topics of this lecture is biosynthesis of lipids, or it's called also lipogenesis. Lipogenesis or biosynthesis of lipids, the main pathway for the de novo synthesis of fatty acids or lipogenesis occur in the cytoplasm or cytosol mean extra mitochondrial system. إذا نتذكر أنه beta oxidation of lipids occur in the mitochondria. Here the biosynthesis of lipid occur in the cytosol. Extra mitochondrial system of fatty acid synthesis or mean the cytosol system of synthesis. This system is present in many tissues including liver, kidney, brain, lung, mammary gland and adipose tissue. It is present in different tissue in the body. Its cofactor requirements include NADPH, ATP, manganese, biotin and HCO3, which is bicarbonate, as a source of CO2. Acetyl-CoA is the intermediate substrate and the free palmitate is the end product. Then this reaction or this lipogenesis need the following cofactor, which is NADPH, ATP, manganese, biotin and HCO3. And in this pathway, the acetyl-CoA is the intermediate substrate, considered as intermediate substrate, and the end product of this pathway is the palmitate, which is an example of fatty acid. Now the sources of acetyl-CoA and phosphate. Sources of acetyl-CoA can be from pyruvate, from amino acids, from fatty acids. All these can give acetyl-CoA. Acetyl-CoA either form stearyl and fatty acid, or can be converted to ketone bodies, or can enter a tricarboxylic acid cycle. إذن من وين بنحصل على الأسيتايل-CoA either from pyruvate or from amino acid metabolism or from fatty acid carboxylation or fatty acid oxidation. الأسيتايل-CoA either enters the Krebs cycle or can convert it to ketone bodies, or it may be used for synthesis of fatty acid and stearyl. ما يعنينا في هذه المحاضرة هو synthesis of fatty acid أو the role of acetyl-CoA أو making it as intermediate substrate for the synthesis of fatty acid. The biosynthesis of fatty acid it takes place in the cytosol. The intermediate should link to acyl carrier protein. Activation of acetyl-CoA should occur. Acetyl-CoA plus CO2 will give malonyl-CoA. The biosynthesis include 4 steps repeating cycle, with each step extension of the chain by 2 carbon atom. يعني كل cycle نضيف 2 carbon atom. The steps involve condensation, reduction, dehydration and reduction. Now steps of fatty acid synthesis. The first steps here transfer of acetyl-CoA from mitochondria to cytosol. The second, activation of acetyl-CoA, and then synthesis of malonyl-CoA. As we said, from acetyl-CoA plus CO2 will give malonyl-CoA. Then elongation cycle of the fatty acid synthesis via acyl carrier protein intermediates. The production of malonyl-CoA is the initial and controlling step in fatty acid synthesis. Bicarbonate as a source of CO2 is required in the initial reaction for the carboxylation of acetyl-CoA to malonyl-CoA in the presence of ATP and acetyl-CoA carboxylase enzyme. إذن احنا نحتاج البيكربونات كsource of CO2 اللي نحتاجه للكاربوكسيليشن of acetyl-CoA حتى تتحول إلى malonyl-CoA. إذا الأسيتايل-CoA will be carboxylated to malonyl-CoA. نضيف له CO2 group. المصدر هذه CO2 هو من البيكربونات. This enzyme اللي هو الأسيتايل-CoA carboxylase enzyme يحتاج إلى الكوفاكتور which is biotin vitamin. إذا يحتاج إلى فيتامين البيوتين ككوفاكتور in this reaction. The reaction takes place in two steps. First of all, carboxylation of biotin involving ATP. Second, the transfer of carboxyl to acetyl-CoA to form malonyl-CoA. إذا الخطوة الأولى هي carboxylation of biotin بوجود ال ATP. الخطوة الثانية هي انتقال الكربوكسيل جروب من البيوتين إلى الأسيتايل-CoA to form malonyl-CoA. And this picture shows how acetyl-CoA converted to malonyl-CoA in the presence of carboxylase enzyme. And as we said, this steps need for biotin as a cofactor and need for ATP. The acetyl group gets transferred from CoA to acyl carrier protein by acetyl-CoA acyl carrier protein transacylase. Next, the malonyl group gets transferred from CoA to acyl carrier protein by malonyl-CoA acyl carrier protein transacylase. Then both acetyl group and malonyl group will be transferred from the CoA to acyl carrier protein. The carboxyl group of malonyl acyl carrier protein then is removed as CO2, and the acetyl group, which contain the carbon number 15 and 16, gets transferred to the alpha carbon of malonyl acyl carrier protein. يعني الأسيتايل جروب اللي هي تمثل الكربون اتوم نمبر 15 and 16 تجي ترتبط مع الألفا كربون of the malonyl acyl carrier protein. This results in 3 ketoacyl carrier protein. إذا بهالحالة هاي راح نضيف الأسيتايل جروب إلى المالونيل جروب ونحصل على الألفا ketoacyl carrier protein. This picture shows the steps of biosynthesis of fatty acids. Here the fatty acids consist of 16 carbon atom, which is palmitic acid. And the first step is condensation of acyl carrier protein with malonyl acyl carrier protein. And this will lead to formation of 3-oxobutanoate. Now the 3-keto group is converted to CH2 by a series of reaction, namely إذا الكيتو جروب اللي تكون عندنا يتحول إلى CH2 group by a series of reaction. The first reaction is reduction to a hydroxyl group in the presence of enzyme 3-ketoacyl carrier protein reductase enzyme. The second by dehydration to form a 2,3 double bond, and the enzyme is needed is 3-hydroxyacyl carrier protein dehydratase enzyme. And third, a second reduction to remove the double bond, and the enzyme needed is enoyl acyl carrier protein reductase enzyme. Then 2 reductase enzyme and 1 dehydratase enzyme is needed in these reactions. Both reduction reaction required for NADPH as a cofactor. إذن الكوفاكتور اللي موجود في reduction reaction is NADPH. The sources of this NADPH that is needed for the reduction reaction is from hexose monophosphate shunt and from the citrate shuttle. إذن هذا هو source of NADPH. So as we said, the 3-keto group undergo the first reduction and converted to 3-hydroxy hydroxybutanoate, and then by dehydratase enzyme, removal of H2O and converted to trans 2 butanoate, and with the other reduction it will convert it to butanoate. The process then continues with the addition of a further 6 units of malonyl acyl carrier protein by successive cycles of these reaction until palmitoyl acyl carrier protein is formed. يعني هذه العملية تتكرر، يرجع مرتلخ المركب اللي طلع عندنا التشين اللي حصلنا عليها، تدخل مرتلخ السايكل اللي هي الكوندنسيشن والريدكشن dehydration and another reduction again 6 with addition of 6 unit of malonyl acyl carrier protein. إذا البروسيس تتكرر 6 مرات، نضيف 6 molecules من المالونيل acyl carrier protein حتى نحصل بالنهاية على البالميتايل acyl carrier protein. At this point, the thioesterase enzyme remove the fatty acyl product as the free fatty acid from the fatty acid synthetase, and it must be converted to CoA ester before it can be entered to various biosynthetic pathway for production of a specific lipid. إذا احنا هنحصل بالنهاية على البالميتايل acyl carrier protein بوجود الثيوستريز انزيم يعمل لنا removal للفاتي acyl group from the acyl carrier protein ويحولها إلى يرجع ينقل هذا الأسايل اللي هو البالميتايل إلى الكو-اي حتى نحصل على البالميتايل كو-اي which enter أو needed أو used for a production of different types of lipid. So as we said, this process will be repeated for 6 cycles, and we need 6 unit of malonyl acyl carrier protein to be added 1 malonyl in each cycle. يعني كل دورة راح نضيف 1 malonyl acyl carrier protein. فهذه العملية تتكرر 6 times أو 6 cycle حتى نحصل بالنهاية على البالميتايل acyl carrier protein which should be converted to palmityl CoA in order to be used for synthesis of other lipid. Now palmitic acid, which is 16 carbon atom, is the predominant fatty acid generated through de novo lipogenesis. Then the predominant fatty acid synthesis by the de novo lipogenesis is a palmitic acid, and the reaction can be expressed as follows: acetyl-CoA with 7 malonyl-CoA plus 14 NADPH plus 14 hydrogen lead to formation of palmitic acid. يعني حتى نكون البالميتيك acid نحتاج إلى acetyl-CoA زائدا 7 malonyl-CoA مع 14 NADPH and 14 hydrogen. Now the fatty acid synthase. Fatty acid synthase is a polypeptide chain with multiple domains, each with distinct enzyme activities required for fatty acid biosynthesis. يعني here the enzymes اللي تسهل reaction in the synthesis of the fatty acid تكون مرتبطة with each other. They present in certain domains and all of them is part of fatty acid synthase. Coenzyme A is an activator of for beta oxidation, while for fatty acid biosynthesis the activator here is acyl carrier protein. And بنفس الوقت هو الأسايل carrier protein is a part of the fatty acid synthase complex. الأسايل groups get anchored to the coenzyme group of acyl carrier protein by thioester linkage. Then condensing enzyme beta ketoacyl synthase also is a part of fatty acid synthase. إذن الإنزيم اللي نحتاجها في البيتا oxidation are separated, while here the enzyme are part of fatty acid synthase. Each of these enzymes present in certain domains in this fatty acid synthase polypeptide chain. Well, this picture shows the fatty acid synthase multi-enzyme complex. So it consists of domains, وكل domain contain certain enzyme needed for the biosynthesis of the fatty acid. Elongation of fatty acid chain occur in the endoplasmic reticulum. This pathway, the microsomal system, elongates saturated and unsaturated acyl-CoA from carbon number 10 upward by 2 carbons using malonyl-CoA as acetyl donor and NADPH as reductant, and is catalyzed by the microsomal fatty acid elongase enzyme. Elongation of stearyl-CoA in the brain increases rapidly during myelination in order to provide carbon chain with 22 carbon atom and with 24 carbon atom fatty acid chain for sphingolipid. إذن elongation يحدث في الendoplasmic reticulum سواء على الsaturated أو unsaturated fatty acyl-CoA. elongation ممكن يكون من chain إذا كانت عدد الكربون ذرات الكربون 10 أو أكثر، elongation يكون بإضافة 2 كربون أتوم اللي نستخدمه ك elongator أو substance حتى نضيف الكربون أو unit حتى نضيف الكربون atoms هي المالونيل-CoA. إذا المالونيل-CoA هو يستخدم أو تستخدم في elongation of the fatty acid as acetyl donor. الريدكتنت اللي يدخل in this reaction هو NADPH. Now the main source of NADPH for lipogenesis is pentose phosphate pathway. احنا قلنا في بداية المحاضرة انه المين source of NADPH is from pentose phosphate pathway اللي هو نفسه الهكسوز monophosphate shunt. إذن NADPH is involved as a donor for reducing equivalent in both the reduction of 3 ketoacyl and the 2,3 unsaturated acyl derivatives. إذن احنا نحتاجه في reduction steps. إذن ال NADPH is used as a donor of reducing equivalent in the reduction reaction. The oxidative reactions of the pentose phosphate pathway are the chief source of hydrogen required for the reductive synthesis of fatty acid. إذن the main source for hydrogen chief source or main source of the hydrogen required for the reductive synthesis of fatty acid is from pentose phosphate pathway. أشنو هو الهيدروجين required for reduction اللي هو ال NADPH. The other source of NADPH include the reaction that converts malate to pyruvate catalyzed by the malic enzyme اللي هي ال NADP malate dehydrogenase. المالك enzyme هو نفسه ال NADPH العفو ال NADP malate dehydrogenase. إذن two source of the NADPH. المين source هو البنتوز فوسفات pathway اللي هو نفسه الهكسوز مونوفوسفات شانت، وأيضا عندنا other source اللي هو الماء from the conversion of malate to pyruvate which catalyzed by malic enzyme or dehydrogenase enzyme. This picture shows the transfer of acetyl-CoA from mitochondria to cytosol, and also show the source of NADPH, as we said, is from the conversion of malate to pyruvate in the presence of malic acid. نحصل على ال NADPH, and from hexose monophosphate shunt أيضا نحصل على ال NADPH, which is important for fatty acid synthesis. Now, as a transfer of acetyl-CoA from mitochondria to cytoplasm, acetyl-CoA cannot pass the mitochondrial membrane. So acetyl-CoA in the mitochondria with the presence of oxaloacetate will form citrate. إذن acetyl-CoA plus oxaloacetate in the presence of citrate in synthase enzyme يتحول إلى citrate. This citrate can pass from the mitochondria to outside to the cytoplasm and يعني citrate can cross the mitochondrial membrane to the cytoplasm. In the cytoplasm, in the presence of citrate lyase enzyme, then citrate will give acetyl-CoA and oxaloacetate. Then oxaloacetate convert to malate and malate to pyruvate in the presence of malic enzyme, and this steps will give NADPH. يعني الأسيتايل-CoA ما يقدر يخترق الميتوكوندريال membrane، يجب أن يتحول إلى other structure or other molecule which is citrate. حتى الستريت ممكن أن تخترق الميتوكوندريال membrane إلى السيتوبلازم ويرجع السيتوبلازم in the cytoplasm الستريت will give acetyl-CoA and oxaloacetate. Hormonal regulation. Insulin is a peptide hormone that critical for managing the body's metabolism. It released by the pancreas when the blood sugar rise, and it has many effects that broadly promote the absorption and the storage of sugar, including lipogenesis. An important hormonal regulation of the lipogenesis is insulin. Role of insulin and citrate. Insulin acts to stimulate fatty acid synthesis by increasing the supply of acetyl-CoA because of increasing glucose utilization. إذا بوجود الإنسولين يحصل عندنا glucose utilization ونحصل منه على الأسيتايل-CoA. فإذا وجود الأسيتايل-CoA هو needed for the fatty acid synthesis. So insulin acts as a stimulant for fatty acid synthesis. This is a point for the interconnection between carbohydrate and lipid metabolism in the body. وهذه هي العلاقة بين الكربوهيدرات والليبيد metabolism in the body. Citrate also stimulate the synthesis of acetyl-CoA and malonyl-CoA which are needed for the fatty acid synthesis. إذا وجود الستريت أيضا يعمل لنا stimulation للformation of acetyl-CoA والmalonyl-CoA اللي نحتاجها في الفاتي acid synthesis. Whereas the end product, which is palmitoyl-CoA, inhibit the pentose phosphate shunt and different key enzymes. So الactivator اللي يزيد عندنا السينثيسيس of fatty acid هي بالانسولين والستريت. الانسولين والستريت increase أو stimulate fatty acid synthesis, while on the other hand end products which is palmitoyl-CoA inhibit pentose phosphate shunt، فما يظل عندنا supply لل NADPH اللي نحتاجه كreductant and different key enzyme also inhibit different key enzyme اللي هي مهمة في الفاتي acid synthesis. It appears that the control mechanism are trying to coordinate the rate of synthesis with the rate of utilization of the fatty acid and with the state of carbohydrate metabolism. طبعا البودي يحاول أن يعمل توازن بين السينثيسيس of the fatty acid والutilization واستخدام هذا الفاتي acid مع الستيت اللي هي حالة الكربوهيدرات metabolism. This table shows the comparison between beta oxidation and fatty acid synthesis. Regarding site, beta oxidation occur in mitochondria while fatty acid synthesis occur in the cytoplasm. The intermediate in beta oxidation present as coenzyme A derivative, while in fatty acid synthesis covalently linked to SH group of acyl carrier protein. The enzymes in beta oxidation present as independent protein, while in fatty acid synthesis multi-enzyme complex. A sequential unit, 2 units released as acetyl-CoA in two carbon unit released as acetyl-CoA in beta oxidation, while in fatty acid synthesis 2 carbon added as malonyl-CoA. المالونيل-CoA هو 3 carbon atoms، لكن احنا اللي قنضيفه بالsynthesis of fatty acid هي 2 carbon atom, as 1 carbon atom is released as CO2. Coenzyme in beta oxidation is NAD and FAD, while in fatty acid synthesis is NADPH. The transport in beta oxidation is the carnitine, while in fatty acid synthesis is citrate. The end product in beta oxidation acetyl-CoA, while in fatty acid synthesis is palmitate or palmitic acid. In summary, fatty acid synthesis occur in cytoplasm. The intermediates are linked to acyl carrier protein. The enzyme of fatty acid synthesis are linked in a complex. Elongation occurs by 2 carbon atoms per cycle. The required reducing agent is NADPH, and in general elongation stop at carbon 16. These two things are required for de novo fatty acid synthesis. First, a source of carbon atoms, which is specifically acetyl-CoA, and source of reducing equivalent, which is specifically NADPH. Thank you very much.